Andy Ferguson from the University of Chicago is our next speaker. Andy is an associate professor and at the Pritzker School of Molecular Engineering, and he's the vice dean for equity, diversity, and inclusion, also at the PME. He received his first and a PhD from Princeton University. Before coming to the University of Chicago a few years ago, Andy was a professor of chemical engineering at the University of Illinois at Urbana-Champaign. His research, as you will see in a moment, specializes in developing new, interesting, powerful algorithms, some of them using machine learning, to understand the assembly of complicated molecules, or, for example, the development of antiviral molecules. Andy?
Thanks very much, Juan. Yeah, thank you for the kind introduction, and thank you for having me here. It's thank you to Juan and Beck and all the other organizers. It's been a wonderful 3 days, and so I really enjoyed myself learning some of the new stuff that's going on. Great. So today I'd like to tell you something a little bit similar to Jen Lisgarten's story about data-driven protein design and some of the work they've been doing there using machine learning and high-throughput experimentation to try and engage this problem. So I think it touched on a lot of themes that Max Welling talked about, that John Kodera talked about, and Jen Lisgarten talked about. So I'll try and make some contact there. Great, so this is the folks I did the work with. So it's in collaboration with my colleague and Professor Rama Ranganathan, who's in PME also, and also in biochemistry, and then 2 very good graduate students, Janran Leon and Niksha Praljak, who's sitting in the back here. And this work was supported by the Camille and Henry Dreyfus Foundation. I just need to make the disclosure that Ram and I have a small startup company called Evozyne, just in downtown Chicago, and I just have to say that the company had no bearing in any of the work that I'll talk about today. Okay, so I think this was motivated really well by Jen's talk the other day, that proteins are fantastically complicated molecular machines, and if we could understand how they're built and how to engineer them, we could then repurpose them for other goals. For example, industrial enzymes or curing disease, perhaps optimizing them things that natural selection has not yet optimized them for, but then we as scientists and engineers can sort of convince them to do some other useful functions. So for example, this is happening today, of course. We have, if you're doing a laundry detergent with some active enzymes in it, you're using things like lipases and proteases that have been engineered to withstand the temperatures of the wash, for example, or lignanases. If we could sort of engineer these things to digest lignin, we could then perhaps sort of use sort of biofuels without the net energy loss that frequently comes from sort of using them today. And then, of course, immunoglobulins. If you could develop new antibodies to target sort of emerging diseases, this could then be a game changer for sort of protecting public health against new emergent strains of pathogens. So if you're not familiar with proteins, here's sort of the 101 intro to a protein. It's basically just a heteropolymer. So it's just a string of little beads, and those beads are known as amino acids. And then this sequence of amino acids in this chain dictates what then the protein folds into, dictates its structure, and therefore its function. And so the sort of, which I will touch on in a second, sort of the central paradigm of protein folding, structural biology is sequence dictates structure, dictates function. And so the rules of the game are basically, can we determine what the right sequence of amino acids we should put in this linear chain to give the protein with the right structure and then the right function that we want? Okay, so this is a very old idea, and so this goes back to sort of the 1970s with Christian Anfinsen and Cyrus Leventhal and others. But the whole concept is that nature designs through natural selection. The protein sequence dictates the structure, dictates the function, and it's this enormous diversity in structure and sequence that allows proteins to perform all of the functions that are essential to biology. And so all the proteins that are working in our bodies all have sort of the similar paradigm. It's just a sequence of amino acids, but they have enormously different functions. They can digest food, they can help us sort of help us grow, they can be signaling molecules, they can be immune stimulating molecules, et cetera, et cetera. Okay, so I think Jen covered this beautifully a couple days ago, but so there are many protein folding problems, and so perhaps the protein folding problem is, can you predict the structure of the protein from its polypeptide sequence, its sequence of amino acids? The inverse protein folding problem, which Jen also talked about, is can you sort of have a structure that you want, perhaps because you know it's going to bind a particular target, and then figure out the sequence of amino acids that will give you that structure. The problem I'd like to talk about today is the protein design problem, which is to leapfrog over the structure and say, can I convince a machine to tell me what sequence I should devise in order to have a protein that executes a particular function. And so because of the central paradigm of sequence to structure to function, the sequence dictates both the structure and the function. So it's legitimate to jump over the structure. We're just going to sort of hop right over that and go directly to the functional design. And that can be beneficial, as Jen mentioned, for places where you actually don't know what structure you want. You know that you have a signaling protein that's involved in some biosynthetic cascade. You don't even know perhaps what its signaling partners are, but you know you would like it to be very active. So in this case, you don't need the structure to actually optimize the protein. Okay, so the first problem, and this is maybe less of a controversial statement than a couple of years ago, is that has been essentially solved by AlphaFold 2 and its progenitors. And so the idea that we can actually now predict for a large class of proteins, absolutely not all proteins, there are certainly edge cases where this is not true, but a large class of sort of single chain proteins, we can now predict to high fidelity the structure from the sequence down to sort of 2 angstrom resolution, which is sort of fantastic. This was sort of a dream just 10 years ago. So how about actually doing the sequence to function design? And so if we skip over the structure and say we want to design a protein with a particular function, how does one do that? So there's a couple of sort of generic models to do that, sort of the 2 monoliths of protein functional design. So one is protein sort of physics, and so to use physics-based models. And one of the pioneers in this field is of course, David Baker, who has worked for a long time on things like the Rosetta software infrastructure, using basically sort of force fields, so-called interactions between atoms. So Max Welling talked a lot about this in just the previous talk, to actually predict what the structure of the protein is going to look like, and then perhaps what it's going to do. Can it sort of bind a ligand? Can it do something useful? And so a lot of this is sort of a physics-based design. You're sort of convincing the computer to simulate what these proteins are doing, what the structure is going to look like, using physics potentials, in some cases augmenting that with statistical potentials, and then sort of iterating on those designs using some sort of smart ML techniques to sort of go around this design cycle. And this has been quite effective in designing sort of very folded proteins, hyperstable proteins, but perhaps has been less successful in designing new functionality, simply because you're going after the structure rather than the function, using the structure as a proxy for the functional goal that you care about. Nevertheless, there have been some enormous successes. So here's a paper that I particularly like from I think it was 2017, from David Baker and Jesse Bloom, working in collaboration to sort of design high avidity proteins to bind the hemagglutinin A spike. And so this was sort of going after what was a very well-defined structural target. We know what the HA spike of influenza looks like, and design proteins that are going to bind that with high fidelity. And so this can then be a potential therapeutic for influenza. And this was shown to actually be very successful in sort of lab tests. And so this is, if you do have a structural target, this is a very good and very powerful thing to do. So how about if you don't have a structural target? So if you actually want to leapfrog over the structure and go directly to the function. And so then the standard approach to use is something called directed evolution. And this is, of course, what Francis Arnold, so a lot of my Caltech colleagues are in the audience here. Francis won the 2018 Nobel for this. And it's a very simple idea at core, but then, of course, the devil is in the details. And so to me, as sort of a computational scientist, this looks like a genetic algorithm in real life. And so it's basically a mutation selection algorithm. And so what one does is start with some protein that maybe has some basal function that you care about. Maybe you have a protease that you want to work at a very high temperature to put in a bioreactor. And so perhaps you go to the thermophilic organisms that you know about and find a protease that works at a reasonably high temperature, but maybe not the high temperature that you actually care about. So you start with that. And then maybe you apply some mutations to it. So you do some random mutagenesis, some crossover, and then you rank order the mutants according to some assay. So then you have to go into the lab and figure out which mutants are the winners, which ones are the losers. And then you pick the best ones, and then you recycle the daughters to the next round of selection. And so you're basically doing mutation, selection, and then recycling. And by doing this multiple times, you can hopefully upgrade your functionality from this basal level by introducing mutations that are beneficial to the functionality that you care about. So there's a couple of deficiencies with this. The biggest one is that this search process is highly inefficient. And so the search space is just phenomenally large. It's literally astronomically large. For a 100-mer protein, a protein with 100 amino acids, there's something like 10 to the 260 possible different sequences. So to put that in perspective, there are 10 to the 80 estimated protons in the observable universe. So it's fantastically larger than that number. So the search space is just gargantuan. And so by searching in this sort of random way, and also this blind way that you're just making random mutants, you're only probing a very small sliver of that search space. And most of your mutants are actually dead. And so you're actually not getting very many productive hits in this process. Nevertheless, you can be very successful with this, and Francis is an incredible body of work on using directed evolution for a lot of fantastic things. Here's just one example of working on a cytochrome C enzyme. this was isolated from this particular organism. And the cytochrome C enzyme, Francis convinced it to sort of do something that's very non-natural, which was to catalyze chiral carbon-silicon bond formation. Nature seemingly has no use to do this, but we as engineers may care about this. And so it turns out this enzyme from the wild type, the natural organism, has some sort of basal activity, so some low turnover, and actually catalyzing this chiral bond formation. So this is the enantiomeric excess. And then after 3 rounds of directed evolution, this was sort of elevated by a couple of orders of magnitude by introducing these mutations. And so this is an amazing sort of gain in this functionality through this sort of directed evolution search process. But what I would like to observe is we're still actually very local. We've only made actually 3 mutations relative to the wild type. So potentially there's lots of room left to run if we're able to sort of supercharge this process to search more deeply in the sequence space. Perhaps we could actually do even better and do it much more quickly. Okay, so people are, of course, aware of this. There are many people working in this field. Jen sort of mentioned it is a sort of very busy field these days. And so I think one of the first places I saw people start seriously articulating this was, again, in this beautiful paper by Francis and Kevin Yang. I'm not going to read the whole thing, but this is just a quote from the paper that I find quite inspirational, which says, only a tiny fraction of the amino acid landscape encodes functional proteins. A method for sampling from the distribution of functional proteins would enable large jumps to previously unexplored sections of sequence space. So really what I think Francis is trying to convey here is use machine learning to help design the proteins. And so if we had some model to sort of guide us through the sequence space, and so could we actually then take big jumps and actually get more shots on goal towards the proteins that are likely to be productive? And so, of course, we know, as everything else in the world is governed by the Schrodinger equation, it turns out that at the mesoscale of sort of protein function, that's not a terribly useful description, and we don't have good analytical models to go from sequence to function. So we need to do something else. We don't have good theories, so we're going to have to do something data-driven, which is really why it's got to be sort of a machine learning problem. So the idea is you can then replace this little chunk in your process with some data-guided model that tells you what mutations to make that are very likely to give you high-performance proteins. And so get many more efficient designs here. Okay, so then what this looks like is then basically a design build test process, which is something we as engineers sort of do all the time. Max Welling mentioned a little bit about this in the design of sort of aerofoils, for example, is that we use our model to design some new proteins. We then actually build them. So building is actually easy. It's gene synthesis. You literally go to Agilent or Twist's website, which looks very much like an Amazon cart, and you just punch in the sequence that you want, and they ship you the genes in the mail, and then you put them in the organisms that you care about and express them. And so it's actually, this has now been commoditized to actually be able to do the gene synthesis for proteins that are not too long. You then need an assay. And so the assay can be anything. And so I have colleagues, for example, Jim Fettner at UW Seattle, who does a computational assay, which is to compute delta Gs of binding. This was something that John Chodara talked a lot about yesterday in his beautiful talk. And so it could be a computational assay, saying we want these things to bind as strong as possible to something we care about. And our work is going to be an experimental assay. We're actually going to go into the lab and measure the thing that we care about. So we're going to integrate over all the things that are important, you know, the environment, how you express the protein, et cetera, and use that as our output. Once you measure these things, you can then recycle, retrain the model, and you have this virtuous design build test cycle, which is essentially just machine learning guided directed evolution. Okay, so I'm going to spend basically the rest of the talk talking largely about part 1, which is how we do this design. So here is the picture that I have in my head when I sort of read Francis Arnold's quote, is that we're really using data-driven generative models to learn sort of nature's design rules, by which I mean the genotype to phenotype mapping that we want to go from what is the sequence of the protein to the functionality that we care about. And so you can sort of think about this as maybe like a pictorial map like this, that perhaps you have a bunch of wild type natural sequences. These are sequences that nature has designed by natural selection over millions of years of evolution. Perhaps you can train a model over that to go from the sequences to the function that we care about. So for example, proteolytic activity. If you train this model, then you can interrogate the model and say, okay, well, can you predict where I should jump next in this high dimensional sequence space? And maybe it could tell you, well, I predict there's going to be some fitness peaks out here with high functionality, maybe another one out here. Here's how you should design your proteins and maybe test these ones. Of course, it's not going to be correct all the time. And the further you jump, probably the less certain it's going to be, but it's going to provide you some guidance such that you're not making a random local search. You're making sort of a directed global search. And hopefully that has some advantages in your design build test process. As I mentioned, we're not using structural information. You can use structural information. I'll return to that at the very end of the talk when I close. Okay, so what sorts of models should you use? And so there are many models one could use. There's been a lot of use of sort of simple kernel models, simple regression models in the past. It turns out sort of data-driven generative models are a very good choice. And so why is this a good choice? And so they have sort of three features that make them really valuable and really sort of well suited to this task. And so number one, we can learn the featureization as part of the training pipeline. And so we don't necessarily need to go in and handcraft features about, how are these amino acids different from one another? How should we sort of distinguish them? The machine learning model can sort of learn that on the fly. So you can just present a very simplified representation. We use something like a one-hot encoding or a physical-chemical encoding, and that seems to work very well. It's nonlinear, and that's important because there's an important phenomenon known as epistasis in proteins, which is basically talking about how the amino acids interact with one another to give rise to the emergent function. And these are typically long-range, multi-body, nonlinear couplings. And so linear models are not very good at mimicking that. And so having a nonlinear model in our sort of nonlinear activations in our neural net, I will describe, basically capture that. And then you're generative. And so then you can actually do the inverse design. So you can use a sort of Max Welling and John Chodera nicely put, sort of operate this thing as a search engine that you can say, I actually want this function. Tell me what sequences I should use to design to actually give me that functionality. And so what kinds of models can you use to do this? And so Ram and I wrote a review paper a few years ago, which is already out of date, of course, but here are some of the models that one could then use to sort of do this deep generative design. And so there are many choices. The one that we're going to focus on is something that's familiar to many, Max Welling in particular, variational autoencoders. And so it's a very well understood, very sort of stable model. Caroline Euler presented some beautiful mathematical analysis of autoencoders, which I found quite inspirational on Tuesday, I believe. And so this is the workhorse model that we're going to use. So I'm always very shy. I've given 2 talks, I think, in front of Max, where I have to explain what a variational autoencoder is. So I'm just going to not look over in that corner of the room when I say this. Okay, so if you're not familiar with variational autoencoder, let me tell you what it is in just 20 seconds. So it's an autoencoder because it's encoding its own inputs. And so it sounds like a terribly useless thing to do, but let me bear with me. So what are we providing? We're providing inputs of sequences, which are just one-hot encoded sequences, to the encoder. The encoder's job is to compress that into some low-dimensional space, and the decoder's job is to decompress that back to the full-length sequences. And so the training sort of a protocol is that you would like the sequences to be well reproduced after you train this thing end-to-end. So you're auto-encoding your own input. So if you're interested in what the loss function looks like, basically this chunk of the loss function is saying, I want you to accurately reconstruct the training data that I give you. There's also a regularization chunk, which basically says, I would like you to provide a nice distribution in this low-dimensional latent space. We typically enforce it to be something like a multi-dimensional Gaussian that's easy to sample from, which is something that's going to be important in just 1 second. So we can train this thing up. And so once you've trained this thing up, if you have adequate training data to stably train this, then what you do is you then throw away the encoder and you just run with the decoder. And so what is happening here? If you've trained this thing correctly, you've been able to learn a low-dimensional representation, a compressed representation of your sequences that contain all of the salient information for the decoder to then regenerate the full-length sequences. So you learn something important about the distribution of these sequences. In particular, what are the correlated patterns of amino acid mutations that underpin all of the members of this training data? So if you provide all the proteases that you can find in nature, the autoencoder is then discovering, what does it mean to be a protease? What are the correlated patterns of amino acid mutations I need to preserve to have proteolytic activity? Once you do that, you can then sample from this latent space, which is like jiggling the design rules and saying, okay, I'm preserving all these correlated patterns, but I'm going to shake the box a little bit. Are there other sequences that also look like proteases? And what you hope is some of them actually have superior activity in your functional goals that you care about. And so then as we execute this design build test cycle iteratively, we can then successively upgrade these activities. And so I'll describe that as we go through the talk. Okay, so now we can sort of throw some random numbers in our multidimensional Gaussian, decode some sequences, and then throw them into the gene synthesis and then the assay pipeline to actually figure out, are some of them even better than the ones that nature designed by natural selection? Okay, so now we need a target system to actually go after to sort of test this thing. And so the system I'm going to talk about is something called the SHO-1 SH3 domain. And so it's a very good test system that we're very well set up to do the assays experimentally on. And so what is this? And so the SHO-1 SH3 domain is a transmembrane protein that exists in Baker's yeast. And its job is to sense osmotic stress outside the cell, basically high salt conditions. If it does that, it then signals through the membrane of the yeast, and the SH3 domain, which is the C-terminal tail on this protein, binds this ligand and activates this signaling cascade that causes the cell to pump out a bunch of glycerol that balances the osmotic stress. So it protects the cell from sort of lysing due to these high salt conditions. So if you have a defective SH3 domain, you're going to die if you're exposed to high salt conditions. If your SH3 domain does work, then you're going to be able to survive a high salt challenge. And so here is the 61 residue SH3 binding domain in red here, and here it is in the structure, and here's this ligand that it binds to. It's sort of this twisted beta barrel type thing. Okay, so then we need an assay to actually measure how well these things perform. And so as I said, our assay is going to be purely experimental. And so here's what it looks like. And so we design our genes using the variational autoencoder. We then go to adjuvant or twist, and then we order the genes, we clone them, we transform them into knockouts of yeast that lack the native SHO1 SH3. We provide the one that we designed on this little plasmid, and then we subject them to these high salt conditions. And so then we say, okay, who's going to make it through these high salt conditions? Who did we furnish with an SH3 domain that works? And who was unlucky and got one that didn't work? And so that's going to be our sort of our test for whether these things function or not. And then we deep sequence on the back end to see who made it through. And so the assay sort of looks like this. We sort of design a bunch of genes, put them into our yeast. We then subject them to 1 molar KCl for a certain period of time, 24 hours, and we see who made it through that challenge. And so what we can do is count at the front end how many counts of gene X existed before the high salt challenge. And then at the back end, we can also do our sequencing and figure out how many of gene X made it through the selection. And so how many of these proteins actually allowed the yeast to survive. You can then take the ratio of these two things. For technical reasons, we take the log, and then we normalize it relative to some null gene that offers very little protection, and relative to the wild type gene, which is the one that nature evolved for Baker's yeast. So the only thing to remember here is that the thing I'm calling the NRE, the normalized relative enrichment, is just a proxy for functionality. So if your NRE is 1, it means you're as good as the wild type yeast. If your NRE is greater than 1, it means you're better than the wild type yeast. So what we're hoping for is to design proteins that have superior functionality, better osmoprotection than the wild type yeast. Okay, so let me show you the results. And so what do? We went to the NCBI non-redundant database. We did a blast search on the SHO1 SH3 from Baker's yeast, and we pulled down 5,300 homologs of this. So these are just proteins that exist in nature that are very similar, according to some cutoff in sort of a similarity to the wild type Baker's yeast. This is now our training data set. So we align them into this nice matrix form that we supply to the variational autoencoder. We train up the VAE, we throw away the encoder, and then we start to interrogate the latent space. So let me show you, first of all, that the latent space is doing something useful. This compressed representation is actually preserving some useful information. So here is the two-dimensional projections of our 3D latent space. And so what I've placed on these projections is basically a subset of the 5,300 sequences for which we have annotations. Annotations meaning we know what these proteins do, or we know where they came from. And so the first thing to note is when you look at these projections, this compressed representation the VAE has figured out for us, you see that to first order, things are arranged by color. And so things seem to be grouped in these colored groups. So what are the colored groups? These are basically the functionality of these proteins. So the red ones are involved in actin binding, for example. The blue ones are involved in NADPH oxidation. So what I didn't tell you was that SH3 domains do other things, not just osmosensing. So nature has used SH3 domains to do other useful things, including some of these functions. And so this is sort of showing you that the VAE, having been exposed to sequence information only, we give it no information about structure or function, has figured out how to group things according to functionality. If you then zoom in into any one cluster, for example, this red cluster right here, you can see there's a hierarchically nested substructure to this where things are organized by shape. And what is shape? Well, this is the organism of origin. This is phylogeny, or the ancestral history. So the circles came from Saccharomycotina, the stars came from Pezizomycotina. And so you can see that actually there's another level of organization the VAE has figured out, which is the historical evolution of these proteins. So it seems to be that the VAE has done something useful in organizing the latent space, which gives us hope we can then use it for sort of the rational design of particular functions. So how do we do that? So then we go in and then we actually assay all 5,300 of these sequences according to that high salt challenge assay, and we quantify how functional they are according to this normalized relative enrichment. So what I'm showing you here is all 5,300 sequences now colored according to the NRE scores, the same two-dimensional projections, and most of the space is blue. So these are all the SH3 domains in the natural world that don't offer osmoprotection. So they're doing other things. They're doing actin binding. They're doing NADPH oxidation. Up in the top left corner here, you see this red hotspot. These are the ones that are involved in osmoprotection. And so this is telling us these are the ones that Baker's yeast and its cousins and its friends are actually using to do osmoprotection. So this is interesting. This now gives us our first toehold to design. It should say if you want to design some new sequences, you should probably start mining this area up here because that's where the osmoprotection is happening. Okay, so then how do we actually do the design? So we did 2 designs. And so the first thing is that we went into the latent space and just sampled globally over the entire latent space. And so this was sort of our baseline. And so we said, okay, we know that most of these things are probably going to have low NREs because they're on the blue side of things. But up here where I've covered over the red cluster, maybe those guys are going to perform a little bit better. So we designed 2,000 sequences just by throwing 2,000 random numbers in this latent space, decoding the full length sequences, ordering the genes, doing the assay. We then also did local samplings. Then we said, well, let's shift our Gaussian. Let's not sample globally. Let's push the Gaussian up into the top left corner and just sample in this red hotspot with the expectation we should do better. We're going to more densely sample the place that we know we have osmoprotection. So we designed around 1,000 sequences using that protocol. Okay, so then we assayed them. And so first of all, let me show you the natural assay. So what am I showing you here? I'm showing you basically the blues and reds that I showed you a couple slides ago are now just shown as a histogram. So on the x-axis is the normalized relative enrichment, so how osmoprotective you are, and the y-axis is just the counts. So this big peak right here, down at an NRE of 0, are all the blue sequences. These are the ones that did not offer osmoprotection. Up here, here's the natural sequences that did offer osmoprotection, the little red cluster that was up in the top left of that scatter plot. Okay, so that's our baseline. This is what nature gave us among these 5,300 SH3 proteins. So then if we look at our global sampling, where we sampled everywhere, very pleasingly, what we see is actually the same distribution. And so actually our synthetic sequences are following the same functional distribution as the natural ones. And this is actually statistically true. You cannot distinguish between these 2 distributions using a Kolmogorov-Smirnov test to any significance. So actually these are truly the same distribution. So this has told us, our VAE has learned us the natural rules of what it means to design an SH3 and can produce the same distribution of functional sequences as nature has evolved over natural selection. So we do note that we do have a slight little chunk here above an NRE of 1. So 29 of our sequences are a little bit better than wild type. Of course, there is some noise in the NRE measurement, so we don't want to read too much into that. But we do see some that have evidence of maybe being a little bit better than the wild type show one. So then we look at our local sampling. And so as we sort of hypothesized, our local sampling did a better job of pushing up to the higher mode of this two peak distribution. So up here, we have much denser sampling of the high NRE mode because we've focused on the top left corner where all the good osmosensors are. And so now we have a much higher probability weight up here, and around 36% of our sequences are actually good osmosensors that are as good or even better than the SHO1 SH3. So then you may ask, well, okay, maybe you're just copying what nature did. Maybe actually these sequences are just very similar to the natural osmosensors. And so let's take a look at actually the sequence similarity. So what am I showing you here? So I'm showing you here on the y-axis a scatter plot of all 3,000 sequences we designed. So the 2,000 global, the 1,000 local, and here is their NREs again on the y-axis. But now on the x-axis, what I'm showing you is the sequence identity to the wild type, the SH3 SHO1. So here at 1,1, one sequence identity to SHO1, 100% identical, NRE of 1. This is where the wild type sequence lives. So what I would like to note is that we see a bunch of sequences above this horizontal line, some that have NRE scores better than the wild type SHO1. We also see a large spread in the X dimension, meaning that we have many, many sequences that are actually quite different from the wild type SHO1. They have sort of very few amino acids in common, up to in some cases little as 40% sequence identity. So this is an enormous jump through sequence space. You're mutating nearly 50% of your protein, but you're retaining functionality. So what this guided sort of a search through the sequence space, the search engine, as Maxwell nicely called it, is allowing you to sort of deeply search the sequence space to find things that are very non-local from what nature has designed. So you may say, well, okay, maybe that's not stringent enough. You had 5,300 natural proteins, not just the one Baker's yeast one. And so if you make the more conservative comparison, looking for the identity, the sequence identity to any one of the natural 5,300 proteins, you still see that we're still mutating something like 30% of our protein, making sort of 19 out of 61 mutations, which would be sort of something that would require many, many rounds of sort of usual directed evolution, where you're just making a few mutations every cycle. Okay, then the next question is, well, where do they exist? So where did you actually make the mutations? And so what I'm showing you here is a space-filling model now. So I showed you that sort of red ribbon diagram of what this SH3 SHO1 looked like a few slides ago. Now I'm just showing you with the sort of atoms actually space-filling. Here's its PXXP ligand that it binds to. And what I'm showing you in yellow are the positions, the amino acid positions that our VAE told us you absolutely have to conserve these things in order to have a functional osmosensor. What I'm showing you in blue are the ones that we knew from the literature before that are involved in ligand binding. These are the amino acids that actually contact the ligand and seem to bind it to this protein to activate that signaling cascade. And then in green, I'm showing you both. So the only thing I'd like to observe here is what the VA has told us. Yeah, there are some residues near this binding site that you need to maintain in order to have your osmosensing function. But there's also these amino acids on the distal side of the protein that are very far from the binding site. They're actually seeming to be critical in mediating this osmosensing function. So this is very non-intuitive. If you're doing purely physics-based design to try and optimize, say, the binding affinity between the PXXP ligand and this protein, probably you'd not be doing a deep search on sort of the backside of the protein. So this is sort of a very interesting question that we don't have full answers to yet, but there appears to be important correlated couplings between these amino acids that are mediating the osmosensing effect in this sort of non-local manner that the VAE was able to sort of figure out for us. So last two slides just to close. And so this is very new results. This is stuff that's just come off the press in the last couple of weeks. So Niksha and Jinran have been busy in the lab doing the next round of this iteration. Everything I told you today was unsupervised. And so we just used our unsupervised learning to learn a latent space and then decode it from it. So now we actually have some functional assays. We can go back in and do semi-supervised learning in the sense that we can now augment our latent space with a prediction head that actually predicts from the latent space what the functionality of these proteins should be, so some subset of the proteins. So actually, I think in Jen's talk, she showed this very nicely, that if you are doing sort of multi-headed prediction from a latent space, you're really then distilling out the salient important information to do sort of multiple things simultaneously. And in our case, what this does is it induces a functional gradient in our latent space that makes it easier to sample from. So rather than having to identify the hotspot, this latent space is now ordered by the functionality gradient, and we can begin to walk up that gradient and begin to sort of design even better sequences with higher osmoprotective function in the next round. So we basically retrain the model using this, then we use some Bayesian optimization to design some novel sequences. And so what we've seen is, I'm showing you again the natural distribution, the global sampling, the local sampling from round one, and now here is our round of Bayesian optimal sampling from the semi-supervised round. Is that what we see is we see a much bigger augmentation of this high NRE peak. They're actually be able to do even better and push even further up into this region of the sequence space where we are getting really good osmoprotection. And so we've moved up from like 50% of sequences with NREs greater than 0.5 to something closer to 85%. So it's encouraging that after you turn the crank twice, you do even better because the model has got even stronger. Now you've trained it over the synthetic sequences and you can actuate multiple improvements. Okay, and then here's just showing that they are again diverse. And so I just want to show that there is sort of X scatter here, showing that these sequences are very, very sequence diverse. So with that, I'll close. And what did I tell you today? I told you one way of sort of learning the functional rules of design, this genotype to phenotype mapping using variational autoencoders, and then using some supervised learning in the second round to do even better designs. So of course, we're also applying this to other protein families, but something we got very excited about recently is exploring sort of alignment-free deep learning architectures. And so this means you can actually train not just over a homologous family, you can train over all proteins in, say, the UniRef database. And what that allows you to do is then transfer learning between sort of the big problem, which is what does it mean to be a folded protein, which I think there is good generic design rules for. So what does it mean to sort of adopt a stable tertiary structure? And then specialize those to your particular functional goal of interest, where that typically, because of the low amounts of data, is more of a niche problem, that every sort of functional design problem is special, whereas perhaps every structural problem is more generic. And so I think we can do some important transfer learning from that. And then, of course, what I've hoped to convince you today, I was sort of inspired by the panel yesterday, what can AI do for science? I think you can almost frame it the other way and say that, it's a big threat not to use AI to sort of do science, that actually it allows us to do things at a scale and a power that's very difficult to beat. And so rather than sticking with the old blind directed evolution, the case I told you today is using AI to help guide our search and supercharge this process to allow us to do things that would not be impossible before, but be very difficult. And so multiple rounds of directed evolution can now be sort of achieved in just a single round of this machine learning guided directed evolution. So with that, I'll close. So thanks again to the organizers for the invite, and be happy to answer any questions.
Thank you, Andy. Aaron.
Andy, you gave this nice example from Francis Arnold of evolving the carbon-silicon bond enzyme. And so that doesn't exist in nature to begin with. So you can't just kind of begin with some enzymes and make them better. So is there a way to jumpstart this whole process in the case of a totally new functionality?
Yeah, it's a great question. So it didn't. It's not true. It did not totally exist in nature. There was one enzyme that had some very low functionality. And so typically, if you can do that, then you can get a toehold. And so then that allows you to actuate this process. So the way we sort of refer to it when that functionality really doesn't exist, you have sort of this cold start. And so in that case, maybe you need to prime the pump by doing something that's more like intuitive design or really screening deeply in, say, a metagenomic database to find something that is close and has a little bit of the functionality you care about. And then hopefully you can sort of upgrade this and get sort of supernatural function. But engineering absolutely non-natural function, that's a much tougher thing to do, as you observe. Absolutely.
Any other questions? People?
Just a quick one, Andy. So I think you motivated the NRE to be greater than 1, right? But a lot of your distributions, you're pegging it greater than 0.5. So even with the semi-supervised approach, it doesn't appear that you're getting many examples actually greater than 1. So what do you think is the next step to be able to actually bias the distribution to where it's even centered at 1 and even much greater?
Yeah, this is a really great point. I sort of glossed over this, but the issue that we have with the current assay, it saturates. And so it's sort of a sigmoidal response. And so we actually don't have great discriminative power between things that have like an NRE of, say, 0.8 versus 1.2, just because the assay is insensitive there. So what we actually need to do is then shift the baseline of the assay to be more sensitive at the higher range or do more detailed characterizations. And so the one thing we're pursuing is a sort of multi-fidelity idea. Actually, Max Welling touched on that in his talk, that you maybe have sort of a fast, sort of a low resolution assay, then you maybe have a slower, higher resolution assay, and you can combine those things together in sort of a profitable manner. And so that's sort of a multi-fidelity learning. And so we have sort of a biochemical assay that is measuring the binding constant. It's just very painful. So whenever I ask Nick or Jenron to do this in the lab, they always roll their eyes because this is sort of a very painful sort of several weeks of work to do. So hopefully by blending those two things, I think we can do something that's greater than the sum of the parts. Thanks.
All right, we'll do one more question. Max?
Thank you for a great talk, and it was really inspiring. You did the VAE, you really introduced very well. Thank you. So I have a question. So is it possible maybe to close the loop before maybe if you have a really good analysis method that predicts the outcome of a test in the easy cases, so you don't actually have to do some of the experiments, maybe with uncertainty on it, so you could sort of accelerate by skipping if you actually can predict the outcome of a test and only do the test when you really, really need to do them. Is that possible?
Yeah, I really like this idea. This is something that we thought a little bit about. And so it was actually, I think, one of Jen's papers we were reading that this idea of introducing some sort of prior to the GPR. So right now we have sort of an uninformed GPR. We just assume that everything has 0 NRE, for example. But maybe you could introduce some smart priors that allow you to do that. So for example, one that Jen had suggested was, are you foldable? And so foldability is then a proxy, is a necessary condition for function. So maybe you could score it with like a Rosetta score and say, well, if you're not even going to fold, then I'm not even going to bother testing you. So that's a very sort of generic thing, but perhaps there are smarter ways of doing that. So this idea of sort of baking in these physical priors in these low data regimes, I think is really important.
You're doing a lot of tests anyway, so you can train from that signal.
Absolutely.
Yeah.
I love it. Thank you.
Thank you, Andy. That was great.